All right, let's dive into one of the most fundamental ideas in all of biology, the central dogma.
You've got three key players here. One flow of information: DNA, RNA, and protein. This simple flow is what we call the central dogma of molecular biology. Really, it's the story of how a cell reads its own instruction manual.
Okay, so let's break that down.
So what is the central dogma really? Well, it's a concept that was first laid out by Crick way back in 1956, and it basically describes the main pathway for genetic information in our cells. You can think of it like this. The information starts with the master blueprint, which is the DNA. Then it gets transcribed into a sort of working copy, the RNA. And finally, that RNA copy is translated to build all the functional machines of the cell, and those are the proteins.
But wait, before any of that can happen, that master blueprint has to be copied, right? This is absolutely essential for when cells divide. And that brings us to our first big process, DNA replication. And the why here is really the whole point. Think about it. Every single time one of your cells divides, that entire genetic instruction manual has to be perfectly duplicated. This is how you make sure that each new daughter cell gets a complete and flawless set of instructions to do its job. So how does it start? Well, the process kicks off with that iconic DNA double helix literally unwinding. It splits apart, just like a zipper, into two separate strands. Now, each of those original strands is going to act as a template, kind of like a guide for building a brand new partner strand. And this is where our star player makes its entrance, an enzyme called DNA polymerase. You can think of it as a high-speed molecular copy machine. It just zips along the template strand, reading the sequence and grabbing the right building blocks, we call them nucleotides, to build the new strand. And it does this with incredible speed and, more importantly, accuracy. Because, that accuracy is critical. Just one little mistake could lead to a mutation. So what's the end result? From that one original DNA molecule, we now have two perfect, identical copies. What's really cool is that each new double helix is made of one of the old strands and one brand new one. It's just a beautiful, elegant system for preserving the code of life. Okay, so the cell has a perfect copy of its DNA. Now what? How does it actually use the instructions locked away in a specific gene to build something? Well, that's where our second step, transcription, comes into play. All right, for this part of the journey, we're deep inside the cell's protected command center, the nucleus. Here, a different enzyme, called RNA polymerase, finds the start of a gene that needs to be read and latches onto one of the DNA strands. Now, instead of copying the entire genetic library, RNA polymerase is much more focused. It just makes a copy of a single page, a single gene. This copy is a single-stranded molecule we call messenger RNA, or mRNA for short. It's basically a transcript of that one gene's instructions. And here's the key part. This mRNA is a portable message. It's like a disposable memo, a copy of an important blueprint. Its whole job is to carry that genetic code safely out of the nucleus and into the main part of the cell, where all the protein-building factories are waiting. And that brings us to the final and honestly amazing step. This is where the message is actually read and the instructions are finally carried out. Get ready for step 3, translation. What's really fascinating is that Crick himself saw a potential problem here. He wondered, how on earth does the cell translate the language of nucleotides into the completely different language of amino acids to build proteins? He had this brilliant idea. He hypothesized that some kind of adapter molecule must exist to bridge that gap.
Okay, so this whole operation takes place on a molecular machine called a ribosome. You can picture the ribosome cramping onto the mRNA strand. It then starts reading the sequence, but not one letter at a time. It reads it in 3-letter words, which we call codons. And wouldn't you know it, here is Crick's hypothetical adapter molecule brought to life. It's called transfer RNA, or tRNA, and each tRNA is basically a molecular translator. One end is built to recognize a specific mRNA codon, and the other end carries the exact amino acid that matches that codon. It's the perfect bridge. So as the ribosome chugs along the mRNA, it reads each codon, grabs the matching tRNA, and links the amino acids together one by one. This creates a long chain. We call it a polypeptide, which then folds up into a complex, 3-dimensional, and fully functional protein. Job done. Now, for a long time, people thought this DNA to RNA to protein highway was a strict one-way street. But, as science always does, things got a little more interesting. We've since discovered some really fascinating exceptions that kind of bend the original rules.
For example, you've got retroviruses, like HIV. They can actually perform reverse transcription. They use their RNA as a template to make DNA, completely flipping the dogma on its head. Then there are other viruses that can just replicate their RNA directly. And if that's not weird enough, you have prions. These are infectious proteins that can pass on information just by forcing other proteins to misfold, with no DNA or RNA involved at all. Wild, right?
Okay, so why is all this so important for a modern field like bioinformatics?
Well, it's because the central dogma gives us the fundamental rules of the game. It's the operating system. If we want to computationally analyze an organism's entire DNA sequence, which is the field of genomics, or all of its RNA messages called transcriptomics, you have to start with this exact flow of information. It's the foundation for everything. Think about a modern technique like RNA sequencing. It completely relies on this process. What it does is capture a snapshot of every single mRNA molecule in a cell at a given moment. This gives us a profile of what we call the transcriptome, and it lets us see exactly which genes are switched on or off. That's how we start to understand how cells work in both health and disease. So, when you boil it all down, it comes back to this simple but unbelievably powerful flow of information. You have DNA, the blueprint. You have RNA, the message, and you have protein, the final functional product. It really is, in essence, the operating system of life itself.
And understanding this flow is pretty much key to understanding life. If you want more explainers that break down complex topics just like this, make sure you subscribe to BioinfoBytes.